Lesson 12, Studying Cells. A cell is the smallest unit of a living thing. Whether comprised of one cell, like bacteria, or many cells, like a human, we call it an organism. Thus, cells are the basic building blocks of all organisms. Several cells of one kind interconnect with each other and perform a shared function form tissue. These tissues combine to form an organ, your stomach, heart, or brain, and several organs comprise an organ system, such as the digestive system, circulatory system, or nervous system. Several systems that function together form an organism, like a human being. Here, we will examine the structure and function of cells. There are many types of cells, which scientists group into one of two broad categories: prokaryotic and eukaryotic. For example, we classify both animal and plant cells as eukaryotic cells, whereas we classify bacterial cells as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, we will first examine how biologists study cells. Cells vary in size. With few exceptions, we cannot see individual cells with the naked eye, so scientists use microscopes to study them. A microscope is an instrument that magnifies an object. We photograph most cells with a microscope, so we can call these images micrographs. The optics of a microscope's lenses change the image orientation that the user sees. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when one views through a microscope, and vice versa. Similarly, if one moves the slide left while looking through the microscope, it will appear to move right, and if one moves it down, it will seem to move up. This occurs because microscopes use two sets of lenses to magnify the image. Because of the manner by which light travels through the lenses, this two-lens system produces an inverted image. Binocular, or dissecting microscopes, work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. To give you a sense of cell size, a typical human red blood cell is about 8/millionths of a meter, or 8 micrometers in diameter. A pinhead is about 2/1000ths of a meter, 2 millimeters in diameter. That means about 250 red blood cells could fit on a pinhead. Most student microscopes are light microscopes. Visible light passes and bends through the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes that undergraduates commonly use in the laboratory magnify up to approximately 400 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the process of enlarging an object in appearance. Resolving power is the microscope's ability to distinguish two adjacent structures as separate. The higher the resolution, the better the image's clarity and detail. When one uses oil immersion lenses to study small objects, magnification usually increases to 1,000 times. In order to gain a better understanding of cellular structure and function, scientists typically use electron microscopes. In contrast to light microscopes, electron microscopes use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification and, thus, more detail, it also provides higher resolving power. The method to prepare the specimen for viewing with an electron microscope kills the specimen. Electrons have short wavelengths, shorter than photons, that move best in a vacuum, so we cannot view living cells with an electron microscope. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, creating details of cell surface characteristics. In a transmission electron microscope, the electron beam penetrates the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly bulkier and more expensive than light microscopes. The microscopes we use today are far more complex than those that Dutch shopkeeper Antony van Leeuwenhoek used in the 1600s. Skilled in crafting lenses, van Leeuwenhoek observed the movements of single-celled organisms, which he collectively termed animalcules. In the 1665 publication Micrographia, experimental scientist Robert Hooke coined the term cell for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses, microscope construction, and staining techniques enabled other scientists to see some components inside cells. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that one or more cells comprise all living things. The cell is the basic unit of life, and new cells arise from existing cells. Rudolf Virchow later made important contributions to this theory. See you in the next lesson.